Hello everyone, Dr. Mungli here. So in this video I will be explaining you about protein digestion process, and also specifically I will be concentrating on gastrointestinal proteases. So my area of interest in this particular video will be about explaining gastrointestinal proteases. Just to give you a brief or a quick review about how the protein digestion is going on in the intestine. So the protein digestion, it all begins in the stomach. So in the stomach we have hydrochloric acid, which will be denaturing proteins and relieving its quaternary structure, tertiary structure and the secondary structure, and you get a polypeptide chain. Now the polypeptide chain that is released, so it will be cleaved into random fragments, polypeptide fragments, based on the specific activity of an enzyme called pepsinogen, which is converted to pepsin in the stomach, mediated by acidity of the stomach. Now, when the polypeptide fragments reaches the intestine, so in the intestine, so further protein digestion will go on using pancreatic proteases and the proteases that are located over brush border epithelium. So the protein digestion, it begins in the stomach, mediated by pepsin activity in the stomach, and further digestion of proteins will go on in the intestine, mediated by pancreatic proteases and intestinal proteases. Now let's move on to see activation of zymogens. So one of the specialty about protein digestion in our body compared to starch and that is a carbohydrate and lipid digestion is carbohydrate digestive enzymes and lipid digestive enzymes, they are already active in their active form, whereas protein digestive enzymes, they are available in inactive forms called as zymogens. So protein digestive enzymes will come in as zymogens and they need to be activated. So the zymogen activation, so it needs a specific chemical or a protein molecule. So let's understand how the zymogens are activated here. So the first enzyme, that is pepsinogen. So note that pepsinogen is coming from chief cells that are there in the gastric mucosa, and these chief cells, they will synthesize pepsinogen, and pepsinogen is converted to its active form pepsin. So activation of pepsinogen into pepsin, it will be mediated by HCl, that is hydrochloric acid, which is synthesized by the parietal cells. So this kind of activation do not need any enzyme mediation, and that is why this is called as autocatalytic mechanism, autocatalysis. So autocatalysis is going on here in the activation of pepsinogen into pepsin. Now the trypsinogen is a pancreatic enzyme, so it comes in from the pancreas into the duodenum, and trypsinogen is activated into its active form trypsin. So activation of trypsinogen into trypsin, this will be done by a brush border enzyme called enteropeptidase. You got to remember this name here, enteropeptidase. So the enteropeptidase is also called as enterokinase. So this is an enteropeptidase or enterokinase. So note that enteropeptidase enzyme or enterokinase enzyme, it is already located over the brush border epithelium of small intestine, especially duodenum. So with the help of enteropeptidase, trypsinogen is converted into its active form, and that is trypsin. Now let's move on to see our third enzyme, chymotrypsinogen. Chymotrypsinogen, which is a pancreatic enzyme, it is converted into chymotrypsin. So chymotrypsin is an active enzyme. So chymotrypsinogen into chymotrypsin, this will be activated by trypsin itself. So the active trypsin, once it is activated from enterokinase or enteropeptidase, that trypsin itself, it's going to activate chymotrypsinogen into chymotrypsin. Now let's move on to see our the fourth enzyme, and that is proelastase. Now the proelastase, it is converted into its active form elastase, and this job is done by again trypsin enzyme. So activation of proelastase into elastase is done by trypsin. Now our fifth enzyme here is procarboxypeptidase A. Now the procarboxypeptidase A is converted into its active form carboxypeptidase A. Carboxypeptidase A, and this activation is done by again trypsin. Now our last enzyme is procarboxypeptidase B. Now the procarboxypeptidase B, it is converted into carboxypeptidase B carboxypeptidase B, and this job it will be done by again trypsin. Now let me quickly recap all the zymogen activation here. So our first zymogen that I told is pepsinogen activation into pepsin done by HCl. Second, and that's a enzyme present in the stomach. Now the second enzyme is trypsinogen, which is converted into trypsin by enterokinase or enteropeptidase, which is located over brush border epithelium. Third enzyme is chymotrypsinogen, converted into chymotrypsin done by trypsin itself. And our fourth enzyme is proelastase, which is converted into elastase done by trypsin. Fifth enzyme is procarboxypeptidase A, converted to carboxypeptidase A done by trypsin. And sixth enzyme is procarboxypeptidase B into carboxypeptidase B done by trypsin. So these are all our zymogens, and this is how they are activated. Now let's move on to see what all the amino acid specificities of these active proteases. Now the active protease pepsin, which is there in the stomach, so note that pepsin is an endopeptidase. It's an endopeptidase. Pepsin is an endopeptidase, and the specificities of pepsin is it will be acting on the carboxyl sides of aromatic and acidic amino acid. Carboxyl side of aromatic amino acids, aromatic and acidic amino acids. That is the specificities of pepsin. So pepsin is an endopeptidase, and it is acting on the carboxyl side of the peptide bonds contributed by aromatic amino acids and acidic amino acids. As you all know that aromatic amino acids are phenylalanine, tyrosine and tryptophan. Acidic amino acids are aspartate and glutamate. So it will be acting on the peptide bond contributed by the carboxyl side of aromatic and acidic amino acids. Now let's move on to see our next enzyme, and that is trypsin. So trypsin is again it's an endopeptidase, and specificities of trypsin is it will be acting on the carboxyl side of lysine and arginine. Lysine and arginine amino acids, carboxyl side. So the carboxyl side contributed by peptide bonds of lysine and arginine, it will be broken by trypsin. That's an endopeptidase. Coming with the chymotrypsin. So chymotrypsin is an endopeptidase. It means it's going to break peptide bonds within the polypeptide. And the specificities of chymotrypsin is it is acting on the carboxyl side of hydrophobic amino acids. Hydrophobic amino acids. It means you need to remember what all the hydrophobic amino acids. Let me quickly give you what all the hydrophobic amino acids. So we have glycine, alanine, proline, and then we have branched chain amino acids, that is valine, leucine, isoleucine. We have proline, and then we have methionine, phenylalanine, and to a certain extent tryptophan. All these are hydrophobic amino acids, and chymotrypsin is going to act on the carboxyl side of all these hydrophobic amino acids. Coming with the elastase. Elastase is an endopeptidase. and it will be acting on the amino acid carboxyl sites of small side chain amino acids. Small side chain amino acids, like it will be acting on glycine, alanine, serine, something like that. So small side chain amino acids, carboxyl side, that's what is the specificity of elastase enzyme. Now coming to the procarboxypeptidase A. Now the procarboxypeptidase A is an exopeptidase. This is something different than other enzymes that we have seen so far. It's an exopeptidase. Now specificity of procarboxypeptidase is, so it is similar to chymotrypsin. It means it is acting on the carboxy terminus. Remember, I am saying about carboxy terminus. Polypeptide chain has got amino terminus and the carboxy terminus. So procarboxypeptidase is going to act on the carboxy terminus amino acid, and if specifically it will be acting on the hydrophobic amino acids. Hydrophobic. So hydrophobic amino acids. So it means at the carboxy terminus, if there are any hydrophobic amino acids, that particular hydrophobic amino acids will be removed by procarboxypeptidase. Sorry, I am not. It is not procarboxypeptidase. It is the carboxypeptidase. So I need to take this pro here. It is the active form is carboxypeptidase. So carboxypeptidase A and carboxypeptidase B. So the carboxypeptidase A enzyme, it is an exopeptidase, and it's going to act on the carboxy terminus of containing hydrophobic amino acids. Now let's talk about carboxypeptidase B enzyme. So the carboxypeptidase B enzyme, just like carboxypeptidase A enzyme, so it is an exopeptidase. So since it's an exopeptidase, it means it is acting on the carboxy terminus. That's why it is called as carboxypeptidase. Specificity is this will be acting on the lysine and arginine. It is just like your trypsin. Only thing is trypsin is an endopeptidase, whereas carboxypeptidase B is an exopeptidase. Specificity is lysine and arginine. So it means if there are any lysines or arginines at the carboxy terminus, at the very end carboxy terminus, that will be removed by carboxypeptidase B enzyme. Now coming with the aminopeptidase. Aminopeptidase is an exopeptidase, just like carboxypeptidase. Aminopeptidase is an exopeptidase. So it means it is going to act at the amino terminus. That's why the name aminopeptidase. So the specificity means it has a broad specificity. It has broad specificity, so it can act on most of the amino acids present at the amino terminus. Then we have intracellular peptidase. So intracellular proteases. So intracellular proteases, again they are exopeptidases. So they will be acting on di and tripeptides, and again they will be having broad specificities. So like this we have all these active proteases here. So let me quickly recap all the things that I have written so far. So we have pepsin that is there in the stomach. So the pepsin is it is an endopeptidase. It is acting on the carboxyl side of aromatic and acidic amino acids. We have trypsin, which is an endopeptidase. It's a pancreatic protease. So it is active on the carboxyl side of lysine and arginine. Then we have chymotrypsin, which is a pancreatic protease. It's an endopeptidase. It is active on the carboxyl side of hydrophobic amino acids. Then we have elastase, is again it's a pancreatic protease. It's an endopeptidase acting on the small side chain amino acids like glycine, alanine, serine on its carboxy side. And then we have other pancreatic protease called carboxypeptidase A enzyme. It's an exopeptidase and it is acting on the carboxy terminus containing hydrophobic amino acids. We have carboxypeptidase B enzyme, which is a pancreatic protease. It's an exopeptidase acting on the carboxy terminus if it contains lysine and arginine. Then we have seventh enzyme, aminopeptidase. Note that aminopeptidase, it is already attached over the brush border epithelium of small intestine especially duodenum and jejunum. This is already available over the brush border epithelium. It is not a pancreatic protease. So aminopeptidase, it's an exopeptidase and it's act on the amino terminus. Then our last enzyme, and that is intracellular proteases. Intracellular proteases, they are exopeptidases. They are already available within the intestine enterocytes, and they will act on di and tripeptides. So these are all active proteases with their specificities and their source, basically from where they originate from. Now as an example, so let's move on to see an example here. So I have given a polypeptide chain for explaining all the enzymes that I have explained so far. So here is a polypeptide chain. So we have amino terminus on this side, that's an amino terminus, and this is the carboxy terminus. So this is the C terminus and N terminus. On the N terminus we have glycine. It means on the left hand side of glycine we have amino group, and the right hand side of glycine, so that's a peptide bond contributed by carboxyl group of glycine and amino group of alanine. So that's the glycine and alanine contributing each atoms there, and that is CONH, and that's the peptide bond. So knowing that, so we have these peptide bonds, carboxy terminus and amino terminus there. So what we will do is we will take example of enzymes and try to break this polypeptide chain into amino acids, individual amino acids, di and tripeptides. Let's see what happens. So what I'll do is I'm going to randomly break this polypeptide chain. So my enzyme of interest is I want to break down. So it all depends on which particular enzyme gets this polypeptide chain first. So as you know, this very first enzyme that acts on the polypeptide chain is in the stomach, because digestion of polypeptide chain starts in the stomach. So in the stomach we have pepsin. So pepsin, as you all know, as I have explained in the previous slide, it is acting on the carboxyl side of aromatic and acidic amino acids. So we have few aromatic and acidic amino acids here. So what I will do is I am going to select pepsin, and my pepsin is going to act on the carboxyl side of phenylalanine. Note that phenylalanine, left hand side there is a amino side, and right hand side is the carboxyl side. So your pepsin is going to break this phenylalanine here. Okay. So with this, if the phenylalanine is broken like that, so you get a polypeptide chain something like this. So now I got glycine, alanine, tyrosine, glutamate, serine, glutamine, aspartate, and phenylalanine. This is what is my fragment now, and that's one fragment, and my another fragment will be, that's plus. Now we have valine, leucine, we have alanine, methionine, glutamate. This fragment is continued here, glutamate. Then we have methionine, threonine, proline, aspartate, leucine, and that is continued as isoleucine. So this is our another polypeptide fragment. Now let's take care of this polypeptide fragment. Consider that this polypeptide fragment gets into duodenum. So in the duodenum what happens? So we have one polypeptide fragment here. So this is one polypeptide fragment, you are one polypeptide fragment, and you are another polypeptide fragment here. So further let's digest these polypeptide fragments. So the digestion of second polypeptide fragment, that is, it can go on something like this. So first let me handle the first fragment, and then I will move on to see the second fragment. So we have glycine, alanine, tyrosine, glutamate, serine, glutamine, aspartate, and phenylalanine. So in the intestine, so we have this chymotrypsin, elastase, carboxypeptidase A, and carboxypeptidase B. So trypsin is specific for lysine and arginine. See if there is any lysine and arginine in the first polypeptide fragment. So in the first polypeptide fragment, so we don't have any lysine and arginine there. So let's move on to chymotrypsin. So trypsin, no lysine and arginine. Let's move on to see chymotrypsin. Chymotrypsin specificity is carboxyl side of hydrophobic amino acids. Now we have few hydrophobic amino acids here. So we have alanine, hydrophobic amino acid alanine, which is interacting with the tyrosine there. So that particular bond, it can be broken. So note that chymotrypsin is endopeptidase, so it can act on the carboxyl side of alanine. Alanine is an hydrophobic amino acid. But one thing that you need to remember here is glycine peptide bond. Glycine is also an hydrophobic amino acid, but chymotrypsin cannot act on here. Right? That's because glycine is the last amino acid and that's on the amino terminus. So amino peptidase has to act on that, not the chymotrypsin. So at the end of action of chymotrypsin, so you get glycine and alanine fragment, and that's one fragment you get, and the rest of the thing will be tyrosine, glutamate, serine, glutamine, aspartate, and phenylalanine. This is what you get at the end of action of chymotrypsin. Now let's see how to further digest this. Let's talk about glycine and alanine here. Now the glycine and alanine both are hydrophobic amino acids. So now there is only one peptide bond. So this has to be handled by exopeptidases. Now the carboxypeptidase A enzyme, it is specific for carboxy terminus hydrophobic amino acids. It means alanine is a hydrophobic amino acid and it can be, this bond can be broken by carboxypeptidase A enzyme. So at the end of action of carboxypeptidase A enzyme, why I am saying carboxypeptidase A means, so this is the N terminus and this is the carboxy terminus for this dipeptide. So carboxy terminus side, from this side, so your carboxypeptidase A enzyme, it can break this and you get glycine individual amino acid and you get alanine individual amino acid. Okay, now let's take care of this particular fragment. So we have a serine here. So serine is a small side chain amino acid and we have elastase enzyme which can clip on the carboxyl side of serine. So elastase can work on that. So you get tyrosine, glutamate and serine. This is on one side, that's one fragment. And then you get glutamine, aspartate and phenylalanine. This is another fragment. Now how to further digest these fragments? So note that, so we have a tyrosine amino acid here and that will be the carboxy side, and serine, this is the amino side. It means this is a tripeptide which can be taken care by exopeptidases. But exopeptidases, we have two carboxypeptidases, that is carboxypeptidase A, which is specific for hydrophobic amino acid, carboxypeptidase B for lysine and arginine. So obviously you cannot use carboxypeptidase A or B for this particular peptide digestion. Now we can use aminopeptidases here, because aminopeptidases, which are located on the brush border epithelium, they have a broad specificity, means they can act at the amino terminus. See, this is the amino terminus. Your aminopeptidase can break this particular peptide bond and give you tyrosine. That's a individual tyrosine can be released. Now you have glutamate and serine. Further down, so aminopeptidase can act on this bond and can release glutamate and serine individually. That's how aminopeptidase can act on. Now let's talk about how to digest glutamine, aspartate, phenylalanine, that particular structure. We have a carboxypeptidase A enzyme, so which is specific for carboxyl terminus hydrophobic amino acids, and phenylalanine is an hydrophobic amino acid. Your carboxypeptidase A can break this particular bond and give you glutamine, aspartate on one side and phenylalanine amino acid is released. Now glutamine aspartate, which is a dipeptide there, so that can be taken care by aminopeptidase from the amino terminus. See this, this is the amino terminus for glutamine, carboxy terminus on the other side, but the amino terminus peptide bond can be broken by aminopeptidase enzyme. So that is located over brush border epithelium. So you get glutamine and aspartate as an individual amino acids. In the same way, so you can go on doing that kind of breakage on the other fragment too. It will take long time for me to explain all this fragment, all this particular fragment, how to digest it. So the principle is very similar to what I have explained for one of the fragment here. You really need to conveniently bring in a specific amino acid. Only thing that you need to remember is endopeptidases, they act on all the amino acids that are within this particular region. So in this particular fragment, endopeptidases will act on these, means they can act on this peptide bond, that is second peptide bond, third, fourth, fifth, sixth peptide bond. So last two peptide bonds in a polypeptide chain, that is the first peptide bond here, that's all the way towards the amino terminus, and this peptide bond all the way towards the terminus. You cannot use endopeptidases for breaking the first peptide bond and very last peptide bond. That will be taken care by aminopeptidases and carboxypeptidases. Other than first and last peptide bond, all the other peptide bonds in a fragment in a polypeptide chain can be taken care by endopeptidases. You really need to note that. Now after that you need to remember specificities of all these amino acids, proteases. So aminopeptidase, it has got a broad specificity. It is acting on the very first peptide bond there from the amino terminus. And the carboxypeptidases, we have two carboxypeptidases, A and carboxypeptidase B. So the carboxypeptidase A will act on the hydrophobic amino acids if they are present on the carboxy terminus. Carboxypeptidase B will act on lysine and arginine if they are present at the carboxy terminus. Now the endopeptidases, trypsin acts on lysine and arginine if they are starting from the second peptide bond and almost towards the last but one peptide bond. In the same way, chymotrypsin is an endopeptidase acting on hydrophobic amino acids. Then we have elastase acting on small side chain amino acid carboxy terminus. Then, so these are some of the things that you really need to remember. And also the pepsin. Pepsin acts on carboxys. It's an endopeptidase. It acts on the carboxyl side of aromatic and acidic amino acids. So that's about it. So I hope this video has helped you to understand how exactly intestinal proteases digest our dietary proteins into almost down to individual amino acids and di and tripeptides. Later di and tripeptides, they can be taken care by aminopeptidases or intracellular peptidases. Thanks again for watching, and I wish you the best for whatever you are doing currently, and see you again in my next video. Till then, take care.